Hey team, Dr. Jekyll here, and now we're going to cover the inflammatory response. So first we looked at how the resident immune cells detect pathogens or damage. Then we detected what signaling they do to induce the inflammatory response. Now we're going to cover the inflammatory response itself, which is super awesome. Okay, so let's jump into it. Yeah, so we've covered the sensing, the signaling. Now we're going to cover the response. Now there's three main kinds of response that we need to cover: the vascular response, the cell infiltration response, and the cell activation response. Now these all overlap, but they roughly happen in that order. The vascular response is first, then the cell infiltration, and then the cell activation. But there's a little bit of overlap, but that's roughly what we're talking about right now. So let's first cover the vascular response. So here we have a blood vessel here. This is the lumen of the blood vessel. On the inside of the blood vessel, we have endothelial cells. And these are really the control cells. Now, on the outside, we have smooth muscle cells, and these smooth muscle cells contract and expand to change the diameter. So this would be an arteriole, which is a small artery, because we know that because it's got the muscle around it. The capillaries and the venous part of it does not have the muscles around it. So this is probably a small arteriole. So let's have a look. First of all, our blood cells are just flowing down it. I know you're thinking, how do I have the budget for such amazing computer animation? But don't worry, that's just the tip of the iceberg. It's going to get better in this presentation. So yeah, the blood cells are just flowing down this arteriole. So on the surface of the endothelial cells, we have cytokine receptors, and this is just one example of them. We have IL-1, that's the inflammatory cytokine, receptor 1. Now there's a few, often each cytokine has multiple receptors, and so we typically just number the receptors. So this is the first receptor of the IL-1 receptor family. So IL-1 receptor 1, and it will be expressed on the endothelial cells. Now, if it gets activated by IL-1, we get the release of nitric oxide. Now, nitric oxide is a smooth muscle relaxant, and actually this pathway is the target of the drug Viagra. So anyway, the nitric oxide causes the relaxation of smooth muscle. But there are also prostaglandin receptors straight on that smooth muscle itself. So activation of that smooth muscle itself can cause relaxation. So what happens when nitric oxide is released from endothelial cells? All the prostaglandin receptors are activated directly on the smooth muscles. We get vasodilation. I know the budget on this is amazing. So we get vasodilation to bring more blood to the site of inflammation. This allows us to have a greater response. It allows us to get more immune cells there. Allows us to get more of the fluid component of the blood that contains antimicrobial compounds to the site of inflammation. And it provides more nutrients for us to do the immune response. Our immune cells become active and very metabolically active. Another thing happens if you activate the receptors. This time we're just looking at a capillary, which is just those single layers of endothelial cells there. So this is just the capillary. This is after the arteriole. If we activate those receptors, we get a breakdown of the tight junctions and we get gap formation in between our endothelial cells. So we get tight junction breakdown after we get cytokine receptors. Another important one is the histamine receptor here that does this too. So the histamine receptor causes that breakdown of the tight junction right there. So why become leaky? I covered this a little bit in a previous video. Why do you want to become leaky? Well, there's a few things. One, the humoral immunity, right? So a component of our adaptive immune system is the humoral immunity, which is antibodies are flowing around our blood. If you get vaccinated for SARS-CoV-2, you will have antibodies to SARS-CoV-2 flowing around your blood, ready to bind to that disease should it be there. So when SARS-CoV-2 gets into our tissues, we need to get a fluid component of our blood in there to help it bind to the virus in the extracellular space, because the virus isn't just going to hang out in our blood system. So it's about flooding the site with humoral immunity, but there's also an innate immune protein almost equivalent of antibodies called complement proteins, and these have an innate ability to bind and kill pathogens, in particular bacteria. These complement proteins, upon detecting a bacteria, can actually self-assemble into a pore forming a toxin for that bacteria to kill that bacteria. So we need to get the complement and the antibodies into the tissue so it can do its job, so we become leaky. Now, we have loads of blood vessels in our nose, for example. So if a whole bunch of fluid then floods into our nasal cavity as our blood vessels become leaky, say you've got a common cold, you're going to end up with a runny nose. So this is one of the major causes of runny noses, is that vasodilation and leakiness of the fluid into our nasal cavity. And this is why, if you have allergic reactions to pollen, your nose also runs too, because histamine can break those tight junctions and allow the fluid to leak out into your nasal cavity. This is actually handy if your nasal cavity is full of viral particles. So it's actually an adaptive response. But yes, a big problem is if we get histamine can be released in response to IgE, the allergy antibody, binding to something that it shouldn't. Sometimes the antibodies can essentially, their binding domains overlap so much between a pathogen and a non-pathogenic substance, or sometimes our immune system can get confused about what is a pathogenic substance and respond to things like pollen or peanuts. Now, should this happen, we get massive histamine release when we shouldn't, and so we get that massive vasodilation and leakiness of the blood vessels when we shouldn't. So I always remember this by allergy antibody doing that. So if that happens in the nasal areas and in the throat areas, that can be really risky for our lives, right? So if you look at some of these air pathways down here, there are narrow parts, partly because of human evolution. We have narrower than the average species because we now walk upright, but we have a lot of narrow parts made out of soft tissue, and when they swell, we can end up with a serious risk to the ability to breathe. So this is where an allergic responses can be incredibly dangerous. It can also be dangerous just because if you make all the blood vessels in your body leaky simultaneously because you have eaten a large amount of peanuts and that those peanut antigens have flowed throughout your body, you can end up with too much leakiness. Your whole body starts to swell, and so your blood pressure drops because all your fluid is rushed out of your vascular system, and your heart begins to pump faster and faster to try compensate for that, and you'll be sweating and your whole body will be swollen. It's an incredibly dangerous situation for that to be happening, right? So the vascular responses can be very dangerous when allergies are occurred, when our immune response is responding to something it shouldn't. There's another thing that happens with our endothelial cells. So should you activate a cytokine or histamine receptor on our endothelial cells, they will start to express adhesion molecules. So these are molecules that dangle out into the lumen of the blood vessel, like this, and they are there to grab immune cells that have a matching molecule that will grab onto the adhesion molecules. So these adhesion molecules are now also expressed in immune cells, and there's sort of a mutual upregulation of both of these adhesion molecules with the response to cytokines. So there are also cytokine receptors on the immune cell there, and their activation will cause the expression of these adhesion molecules. So this causes the immune cell to slow down and eventually stop and adhere to the cell. To the endothelial cell wall, to the vascular wall. And the reason is that they need to stop. They're flowing really fast. If you ever watch a video of the blood, of the vascular system, it is a fast moving machine. And so they need to slow down to adhere, to then grab on and squeeze themselves through the gaps in the endothelial cells to get into the tissue. So this is a really cool video. Let's hope it works. Here we go. So I'm just going to pause it so we can talk about what's happening here. So here, this is a cell culture situation, and on the surface of the plastic, they have adhered endothelial cells, those blood vessel cells. And then flowing over the top are neutrophils. So the really bright cells that are flowing at a high speed are being flowing intentionally across the endothelial cells, and those are neutrophils. Now, what they're going to do is add histamine to the cell culture to see what happens, right? So let's have a look. Here we can see the neutrophils after 1 minute are flowing across incredibly quickly. You can see them flowing past. And now what you can see is some of them start to roll. Now it's actually one group of molecules that allow the rolling and the slowing down, and then another group of molecules that essentially makes them stick and adhere really quickly. So now after 5 minutes, you can see it's a very quick response. The neutrophils aren't flowing across anymore. They're starting to really slow down, and many of them are starting to stop and fully adhere. And so this is what's happening in your blood vessels live, and it's so cool that we can research it outside the body. What are we going to see? Yeah, so that's about the end of the video. So what we can see is we can see the what's happening with these adhesion molecules there. So once we get adhesion and adherence, then chemotaxis takes over. So once I've squeezed through the blood vessel and they're now stopped and they have migrated across the blood vessel, now the chemoattractant effect of chemokines takes over. So the cells are in the tissues and they can now crawl to the site of tissue damage or infection. Now I've got a really cool video on this. So let me break this down for you. So in the red, we have cells that have died, right? So we have tissue damage cells in the red. So here we're looking at a tissue and they've been fluorescently labeled. So these are dead cells over here, and you can see that there's a lot of dead cells around here. The blue is labeling endothelial cells. So here we can see a blood vessel running along here, and the green is neutrophils, the main innate immune cell that responds during inflammation. Most of your white blood cells in your body are neutrophils, and they are the first to be there during an inflammatory response. So in the green, we have neutrophils. Now let's watch this video, which is happening live in a transparent organism called a zebrafish, which allows us to view this response live. This is super cool. So here we can see, oh my gosh, it's a swarm. So this is happening over the course of maybe an hour. Let's just re-watch that. I could watch that on loop over and over again. So you can see here we've got the dying cells. You can see the immune cells are just stopping and pouring across the blood vessel. So they've stopped and they've poured across the blood vessel. Now the chemokines have taken over and they're heading up the concentration gradient in response to the immune cells that will be releasing chemokines around these damaged cells that will be releasing DAMPs. And what you can see is actually them focus on the dying cells, and they will actually phagocytose the dead and dying cells. Isn't it such a swarm? So this is the inflammatory response happening live under a microscope in a semi-transparent organism called the zebrafish with fluorescent labeling. If that doesn't get your love of science going, I don't know what will. That's so sweet. All right, so an interesting question there is, here's a picture of a neutrophil, and they have very funky nuclei. So I want you to stop and think. Oh, I'm bumping the camera. Oh, that's so unprofessional. Okay, yeah, here we go. Why would the neutrophils have those funky nuclei? And the answer is it helps them squeeze through the tiny gaps in the endothelial cells. They can squeeze through phenomenally tiny gaps to get to the other side. A little bit like an octopus, if you've ever seen that. Octopus do it. They do it bit by bit. So they kind of squirt their cytosol through, and then they might pull a lobe of their nuclei through, then another lobe of their nuclei through, then another lobe of their nuclei. So it allows for them to be those first responders. Super cool. Now, let's talk about the neutrophil. The neutrophil is an amazing cell. You're really going to like the neutrophil. Now, there's a lot of cytokines. So when I mention a cytokine, they're going to be the important ones, right? So these are the keystone cytokines that are definitely worth learning. So IL-8 is one of those. So IL-8 is a cytokine. It binds to the IL-8 receptor, and this activates neutrophils. Now, neutrophils contain granules, and again, neutrophils are low in calcium, so when calcium influxes into the neutrophil, that signals for the fusion of the granules to the extracellular, to the cell membrane, just like a mast cell. This is a really common response. Calcium comes in, your vesicles fuse to the cell membrane and release their contents, right? So IL-8 causes this calcium influx, but also bacterial toxins will do the same thing. So it's a great landmine. It's great. I'm going to activate no matter what you do to me kind of situation there. It's kind of a little bit like pulling the pin on a grenade and saying, you shoot me, I'll let go of this grenade and then it will go off. So if you kill the neutrophil, calcium will come in and its granules will be released. So it's kind of like a fail-safe. There's nothing you can do to me, pathogen, because I will release my contents. What's in those granules? Well, in those granules are non-specific toxic compounds, so cytotoxic proteins and compounds designed to kill pretty much everything. One example of this is neutrophil elastase. This is a protein and an enzyme, and it's packed into those granules. And neutrophil elastase is a non-specific protease that will chop up everything. So it kind of will kill the bacteria, but it will also kill host cells. It just chops up everything. It says everything in this area needs to die, and then we can kill everything, and then we can do repair processes later. But it's most important to kill this pathogen. Another enzyme in there is myeloperoxidase, or MPO. Just remember the acronym there. MPO is an enzyme in there. It does a very cool thing. It creates bleach. Hypochlorous acid is bleach, so this enzyme creates bleach. Now, bleach is an incredibly reactive molecule. It's a reactive oxygen species. And so reactive molecules react with other molecules, right? And there's a key kind of principle here, right? The function of a molecule depends on its chemical composition, right? Its chemical composition dictates its structure dictates its function. If you change its chemical composition, you will change its structure and you will change its function. So if you put a very reactive molecule into the extracellular space and it will react with all the molecules out there, it will make those molecules not functional, because it will react with them, changing their chemical composition, changing their structure, changing their function. A good example of this is it could react with phospholipids. Those lipids then stop acting as a barrier. In fact, they start to become less hydrophobic, the lipid component of the phospholipid. So they start to sort of break down and stop functioning as a barrier, and they become permeable, right? And so that allows the cells, that essentially kills the cells. So this enzyme, which creates this reactive oxygen species, hypochlorous acid or bleach, kills everything in the area. And it does that for the same reason why we use bleach on the kitchen sink. It sterilizes the area. So these are two very important proteins that do non-selective toxicity to kill everything in the area, to sort of reset the situation. But here's another thing neutrophils do. If you didn't think neutrophils were cool, you will after this, right? So these are my neutrophils. These are literally my neutrophils, and they're undergoing a process called NETosis. Now, I think I'm just going to describe NETosis. So NETosis is a process where the granules in the neutrophil, instead of fusing with the cell membrane, releasing their content, fuse with the nuclear membrane and infuse their DNA with those bactericidal proteins, those pathogencidal proteins like myeloperoxidase and neutrophil elastase. So instead of MPO and neutrophil elastase going out into the extracellular space, it fuses with the DNA of the neutrophil. Why would it do that? Well, then the neutrophil signals for itself to explode. So the neutrophil then explodes in a process called NETosis. And it's a form of cell death where the neutrophils explode. So if we have a look here we have neutrophils, and in blue we have stained the DNA. So let's watch what happens. Let me choose this guy. Let's choose this guy here. So we can see the DNA, and we can see that the cell was just sitting there. I think I chose the slowest cell to undergo this process. Oh boy, did I choose the one that's going to take the last one? Is it even going to go? Oh my gosh, I chose one of the cells that doesn't happen to. Okay, let's have a look at this cell right here. Hopefully it happens to this cell. We can see the blue DNA. It's jiggling around a bit. Oh, please go. Please go. I can see all the other cells are going. Okay, the DNA is now leaked out into the cytosol, and then it's going to... No, I chose another one. Okay, let's choose this guy. He's going to go. He's going to go. Okay, DNA's leaked out into the cytosol, and now the cell is going to do it. Pop. Please pop. Please pop. Unbelievable. I saw one over here. Okay, so let's watch this guy. Here's the DNA. Here's the DNA. I can't believe I chose 3 that didn't pop. The DNA is now in the cytosol. See how it's kind of spread out? It's not concentrated anymore. We can see the membrane's barely holding on here. And then it will eventually pop. boom, the neutrophil pops. So why would it do this, right? So now we've stained the DNA. Here's the neutrophils, and we have stained the DNA. You can see the DNA is spread out like a net, and this is a neutrophil extracellular trap. So basically what's happened is the neutrophil has coated its DNA in pathogen-killing compounds, such as myeloperoxidase, which is an enzyme, and neutrophil elastase, which is an enzyme that kills bacteria, it kills fungus, and it kills viruses, right? So it has loaded its DNA with that, and then it's going to shoot its DNA out to act as a net to trap the pathogens. Now, this has multiple effects. It prevents the pathogens from going anywhere. It keeps the pathogen-killing proteins at a really high concentration around the bacteria. It's now a net of death, and it kind of limits the collateral damage, because now the neutrophil elastase and myeloperoxidase isn't perfusing throughout the tissue. It's staying locally around the neutrophil, where the neutrophil killed itself, and it stays around the bacteria. It's kind of like hurry-curry. It's absolutely amazing. They've committed suicide to use their DNA, one of the most complicated molecules in the world that contains all the information of life. They're going to crudely use that DNA as a net to trap the pathogen and kill it, because they've laced the net with pathogen-killing proteins. It's unbelievable, this thing. So neutrophil extracellular traps is one of the coolest things of the innate immune response, and this is why the innate immune response is so much better than the adaptive immune response. Think about that. It's flippin' sweet. Anyway, here are these nets. And in blue we have nuclei of parenchymal cells, and in red we have extracellular DNA that is spread throughout the tissue, trapping pathogens. Over here, I've treated my immune cells with Staphylococcus aureus, and that's why they're undergoing that NETosis. So an interesting question is, why would they not degranulate and undergo NETosis? Basically, short, punchy, high concentration IL-8 signaling will induce degranulation, but sustained inflammatory signaling will induce NETosis. The neutrophils will think, okay, what we're doing right now isn't working. The bacteria haven't died. The signals are all still there. We need to take this to the next level, and the next level is neutrophil extracellular traps, or NETs. Super cool. Okay, so here's a question, right? Neutrophils are putting out an enzyme that pumps out bleach, and also putting out an enzyme that digests everything, neutrophil elastase. Isn't that going to cause tissue damage? And the answer is yes. This is a famous study from the late 70s, I believe. Oh, I'll have to look it up. Either early 80s, late 70s. And what they did was they took two groups of mice, and they made one group of mice have no neutrophils. How did they do this? They put in an antibody that binds to the neutrophils, and those antibodies naturally clump and kill those neutrophils. So you can get antibodies from other organisms to bind to the neutrophils of this organism. Very, very clever. It's not really hugely important. Basically, they have a mouse that has no neutrophils, and they have a mouse with loads of neutrophils. Then what they did was, in a sterile setting, they cut a piece of the skin off. They used pain relievers, don't worry, and they had general anesthetics and everything like that. So they tried to be as ethical as possible. It's still the 70s, not hugely ethical. But anyway, under sterile conditions, they cut a small piece of skin off, a tiny little biopsy, to see what would happen. Then they looked at how that skin healed. Now on the left here, after a few days, they had a look at the healing system here. So this is an H&E stain. It's just a histological stain of the wound here. Now what you can see is all these small blue dots here, all these tiny little small blue dots there is neutrophils. So neutrophils have smashed themselves into that tissue damage site, and they've released their neutrophil elastase and their myeloperoxidase, their MPO, and that's caused a whole bunch of tissue damage. Then if you look at the surface here, it's actually still a scab. There's no barrier formed here. This is not healed, right? It hasn't healed yet after this many days. It's still pretty much an open wound full of neutrophils. Over here, this is the mouse that had no neutrophils. And what we can see is we've already seen an epithelial layer form here, right? So this is the skin, this is an epithelial layer, and underneath we've just got a little bit of scar tissue, and that scar tissue will go away eventually. And so this is what healthy skin normally looks like. And so we can see a bit of scar tissue in here. But this is way further down the healing process than this one. We've still got an open wound, no epithelial layer formed on the skin, and it's packed full of neutrophils. Here we've got an epithelial layer. So it seems like neutrophils had slowed down the healing process, and they have, right? Because they come in and they nuke the place with these non-specific cytotoxic compounds. And so neutrophils have actually been associated with worse diseases. So here's Alzheimer's disease. This is a slice of a brain here. And in Alzheimer's disease, we get death of the neurons. So here we have a healthy human brain, and here we have an Alzheimer's diseased brain. And we can see that it's atrophied. The tissue has shrunk away as neurons have been killed. Now, if we look in the brains of Alzheimer's patients versus control patients, and we can use scanning technology to track if inflammation is going on in the brain. So here is an Alzheimer's brain there, and we can see that the red means inflamed. We can see that there's massive inflammation going on compared to an aged matched control. Now, in the elderly, their brains are a little bit inflamed anyway, so there is a little bit of inflammation going on in the control, but way more going on in the Alzheimer's disease individuals. And so this is a memory test in mice, right? So Y-maze is a kind of memory test. It's a very clever memory test here. So here's our control animals. These guys don't have Alzheimer's disease, and they perform very well in the Y-maze. Here we have Alzheimer's mice. They're performing very poorly in the Y-maze because they have a very poor memory, because many of their neurons have died. Their brains aren't working as well, and this is why it's a dementia. Alzheimer's dementia is a really terrible disease that we got to find a cure for as soon as we can. So these guys did the same experiment as the skin guys. They depleted the mice of neutrophils and said, let's see what happens. If we remove the neutrophils from these mice, do we prevent Alzheimer's disease in these Alzheimer's mice? These mice have been genetically altered to get Alzheimer's disease. And what you can see is without the neutrophils, they perform just as well as the control mice. So it seems like neutrophils and the inflammatory response are a core part of things like Alzheimer's disease, and those processes of producing bleach in the brain and producing enzymes that digest up everything like neutrophil elastase are damaging and kill our tissue. So the neutrophil, you might sit back, and if you're Darwin, you might be like, why are you doing this? This is not helpful in any way. You're contributing to dementia. You're slowing down the healing process. Why though? What are you doing? Why do what you do on neutrophil in evolution? So I want you to go back to caveman times. This is actually a picture of Bigfoot. I love Bigfoot. I don't believe he's real, but I just wish he was. I really, really hope he was. I love Bigfoot. Anyway, so this is Bigfoot, but let's imagine he is a proto-human. He's somewhere in human evolution when we were much more ape-like. He gets a cut on his hand. That's going to be the release. That's going to cause the release of damage-associated molecular patterns, right? That's going to cause the release of DAMs, that cut. Now, the immune system has two choices. Do we just mildly respond because we only had the presence of DAMs and clean up the damage and then begin the repair processes? Or do we respond as if there is an infection and spray the area in bleach and spray the area in neutrophil elastase to kill everything and then begin the healing process, right? It has two choices. Now, all throughout human history and evolutionary history, a cut will commonly associate with pathogens getting into the tissue. Remember, the skin is actually an important part of the immune system. It is a barrier, right? And as soon as you cut through the skin, you allow pathogens into there. It is better to over-respond and induce bleach release and neutrophil elastase release and those neutrophils to come packing in, it is better to over-respond, because then you will kill all the pathogens and you will just have a slightly delayed healing response. You might get the formation of a scar, but at least a pathogen hasn't got into your bloodstream, spread around the body, caused sepsis, and died. Now, throughout most of evolutionary history, pathogens have been a major source of human death or animal death. And so by over-responding, you are preventing death, but you are causing a bit of extra tissue damage. By under-responding, you might not cause that little bit of tissue damage that you don't want, that bit of excess tissue damage. You might not cause that, but you might allow a pathogen which will then eventually kill you, and then you won't go on to propagate and spread your genes further down. So animals have evolved essentially to over-respond to pathogens, because it's better to over-respond and cause tissue damage than it is to under-respond and die, right? So our immune system is sort of dialed up to 11. It's designed to over-respond, and it's designed to respond even without the presence of pathogens. So in the case of that dementia, Alzheimer's dementia, you might just be getting neurons dying, releasing DAMPs, and so you actually have no need for your neutrophilic response to occur. All you need is macrophages to eat those dead cells and begin the repair processes. But the neutrophils come in anyway, and they release their pathogen-cidal chemicals that are non-specifically cytotoxic, so they start to kill more neurons. And you can actually see a little bit of a cycle going on there. As those neurons die, they'll release more DAMPs, and so on and so forth. So we end up with that chronic inflammatory situation, which is damaging to the brain, and it can contribute to Alzheimer's disease, even though there are no pathogens present. But evolutionarily speaking, getting a cut and responding appropriately is more important than dementia when you're 80 years old and you've already successfully bred, for example. So that is why we over-respond. That's the answer to Darwin's question. And it's so important to always think of evolution as you are thinking about biology in general. Okay, up next, how do you turn all of this off? This does sound quite damaging and kind of a little bit scary. We need to be able to turn this off. So that's up in the next video.